Hi, everyone. This is episode 741 of People Behind the Science. I'm your host, Dr. Marie McNeely, and today I am thrilled to be sharing my conversation with Dr. Ariel Furst. This episode is made possible with support from our sponsor, New England Biolabs. Listeners, are you curious how molecular biology is moving forward around the world? Hear from the scientists who are driving discovery and expanding applications of existing technologies on the New England Biolabs podcast, Lessons from Lab and Life. Host Lydia Morrison interviews revered scientists working in genetics, synthetic biology, diagnostics, molecular cloning, and other fields. Get ready to explore the cutting edge of science and learn what these remarkable scientists have to offer. Don't miss out on this opportunity to hear exclusive stories about the world of laboratory research. Tune in to Lessons from Lab and Life today, wherever you listen to podcasts. And today, Ariel and I will be talking more about her life and science. So get ready to meet another one of our remarkable people behind the science.
Every day, discoveries are made that will change our understanding of the world around us. Dr. Marie McNeely is here to bring you the brilliant minds who are making these discoveries so they can share their incredible stories and take you on an amazing journey. Welcome to People Behind the Science.
Hello, everyone, and welcome to People Behind the Science. Today, I am thrilled to be speaking with our guest scientist, Dr. Ariel Furst. So, Ariel, welcome to our show today. How are you?
I'm doing very well, thank you, and I'm very excited to be here.
Ariel, we are so excited to have you with us today, and we're looking forward to learning more about your research in our conversation. But first, let me start by giving our listeners a little bit of background. So listeners, Ariel is the Paul M. Cook Career Development Professor in the Department of Chemical Engineering at the Massachusetts Institute of Technology, or MIT. She received her bachelor's degree in chemistry from the University of Chicago and her PhD in chemistry from California Institute of Technology, or Caltech. Ariel was awarded a Beckman Foundation Postdoctoral Fellowship, and she conducted postdoctoral research at the University of California, Berkeley, before joining the faculty at MIT in 2019. Listeners, Ariel has received a variety of awards during her career, including the NIH Director's New Innovator Award, the Camille Dreyfus Teacher-Scholar Award, the Marion Milligan Mason Award from the American Association for the Advancement of Science, the Women in Chemical Engineering Rising Star Award from the American Institute of Chemical Engineers, and the Outstanding Mentor Award from the MIT Undergraduate Research Opportunities Program. She was also named a Scialog Fellow for Negative Emissions Science. And Ariel, today we're excited to hear more about your science, but we also want to hear more about you. So can you start by telling us what you like to do when you're not doing science?
Yeah, I spend a lot of time cooking. I find it very relaxing and in a lot of ways like chemical experiments that are much harder to mess up. Mm-hmm. And I enjoy being outside. So hiking, jogging, all of that good stuff. And I spend a lot of time with my students. We are a pretty social lab, so we do a lot of activities that are pretty Northeast friendly, like apple picking and things like that.
Oh, I love that. I've never been apple picking myself, but it's on my bucket list someday to go.
Definitely. You have to come up. It's an event.
That sounds amazing. And you mentioned you like to cook. Are there particular kinds of food that you're drawn to?
Pretty much anything. We are pretty adventurous in the kitchen, my husband and I, so we tend to just try as many new recipes as we can.
Well, it sounds amazing. It sounds like you've got some fun hobbies to keep you busy outside of science. But I'd love to talk about your scientific side as well. You said sometimes you blend them in terms of doing fun things with your lab members. But let's talk about the research side of your lab. How do you describe your work, Ariel, to people who aren't familiar with your specific field, or maybe people who are outside of science altogether?
So I would say we do very diverse research, but it all centers on energy equity. So this is a concept that's more in social science, but it's really the idea that pollution has disproportionately impacted already marginalized communities. And I feel like it's my job as a scientist to develop new technologies that are accessible to people who really haven't had access to technology before, but who are impacted negatively by it. And we also try to develop tech for energy justice, which is, to remediate those prior harms. So in my lab, we do this by looking to nature. So we do a lot of work with microbes, things that you find in soil. So if you go out in your backyard and get a cup full of soil, you'll probably find some of the things we engineer in our lab. And we do that for projects in human health, in environmental remediation, and in sustainability.
I love that. And I think, Ariel, working towards these big goals of equity and justice and sustainability can be inherently motivating and inspiring. But I think even the best of scientists have some tough times that they have to work through. And I think having motivational quotes or little inspirational bits that you can draw from can help you get through those difficult times. So do you have a favorite quote or a saying or just something in general that really motivates you?
Yes. So we have this actually on the wall in our lab. If you try it once and it doesn't work, try it again.
Mm-hmm.
If you try it a second time and it doesn't work, try it a third time. And if it doesn't work a third time, go take a break and head home for the day.
I like it. Is this something that you implement in the daily lab life?
Definitely. And we also use that if you're, having a rough day and you break some glassware or something. Once you hit 3 times, it's time for you to go home and, watch some Netflix and take a break.
I like that. And I think it is important to step away sometimes, because sometimes you just need to refresh, come at it with a clear mind and try again on a different day, exactly like you described.
Definitely. And I definitely encourage my students to do that as well. We have a book club where we usually read fiction just to make sure that, everyone can step out of science reading. I know when I was a grad student, it was hard to find time to read things that weren't science papers. So we try to intentionally spend some time being creative.
I love this idea of a lab book club, and I believe you're the first one to have one of these that I've heard of. So do you have a favorite book that you like to recommend all of your students read, or that you want me and our listeners to check out, whether it's related to science or not?
I think the most popular in our lab for a kind of science relevant book is Bad Blood about Theranos. We do a lot of diagnostic work, and so it's a nice kind of introduction to some of the pitfalls for the students. And then kind of in the non-science realm, everybody has really enjoyed Crying in H-Mart, which is a memoir about a woman's relationship with her mother and kind of finding herself. And a lot of students I know have read it again since we did it for book club.
Well, these are 2 phenomenal recommendations. I will add them to our website for our listeners to find. And I think in addition to kind of taking a step back and giving your mind a break, I think talking to people in science can help you stay motivated and inspired. So are there people who you've encountered along your career path, Ariel, whether it's mentors or role models or coworkers and colleagues, who've really inspired you and maybe served as role models or mentors?
Yes, definitely. So I actually really didn't like chemistry in high school. I, was not a fan of classes. And when you do lab classes, especially, you kind of know what is supposed to happen, which is the opposite of research. And it kind of takes the fun out of it. And so before I knew what research was like, I was able to spend a few weeks in Shelley Mentier's lab when she was at St. Louis University as a high schooler, actually. And that was really what made me fall in love with science. So she has been a mentor to me since I was a junior in high school. She's still a mentor to me and an amazing researcher. And she's just an inspiration for me.
Oh, that is awesome. And I love to hear these stories of people who are sort of the unlikely version of the scientist they ultimately became, because I think this is something where a lot of people think you have it all figured out from the beginning. So can you take us back to the beginning, Ariel, and maybe these early days when you started thinking about science or getting interested in science?
Yeah, definitely. So, as I mentioned, I think high school was the first time I really saw what science research looked like and not knowing the outcome. And that was very fun for me in a way that I hadn't experienced before. So I started my undergraduate kind of education at Wesleyan University, and it's a fantastic school, but I realized very quickly that I enjoyed doing research in an environment where there were a lot of grad students, which they didn't have. So I transferred to University of Chicago. And, I'll just pause there to say, I think sometimes it can be scary when your original plans don't work out. But I think for me, at least, that's been the most fun part is, learning new things about myself from these experiences. And so I went to U Chicago and I got to work in a lab that kind of solidified my excitement about science. I worked with Steve Kent, who basically was the first one to figure out how to make proteins using chemistry. So, that was the first time I really saw this fundamental connection where chemistry can be used to kind of do whatever you want with it, even, manipulate biology. And he was a fantastic mentor as well. He met with every undergrad in the lab weekly, even though he's, incredibly famous and has done really groundbreaking work. And I think seeing how much he enjoyed still doing the kind of hands-on day-to-day experiment planning with us was what really kind of set me on a path to become a scientist and a researcher.
Oh, I think that is awesome. And I think making these sort of pivots in your career path can be stressful. So how did you make that decision, or was it difficult for you to sort of ultimately pull the plug and say, okay, I have to leave Wesleyan, I have to go to a different institution?
I think it was challenging, but I really, at that point, knew that I really enjoyed doing research and I wasn't finding the right home for that at Wesleyan. So, it was an amazing school and I am still friends with a lot of the people that, were in my dorm hall my first year. But I think in terms of finding research that really motivated me, that was my reason for looking at UChicago.
Well, then it sounds like you were sort of getting set on that path at that point to become the career scientist. What were some of these key moments along the way that really helped you get to where you are today, Ariel?
Yeah, I think, one of them was starting research in high school. And then I think the second was hearing about research going on in the person I ended up working for my PhD, Jackie Barton's lab. I heard her give a talk when I was an undergrad, and her work, again, was one of those kind of mind-bending moments, like when I heard about chemical synthesis of proteins. So she has spent her career looking at how DNA, the stuff that, central dogma makes all the RNA and proteins in our bodies, codes life, how that structurally acts like a wire, which is crazy to me. And the metaphor she used, which hopefully will still resonate with people today, is it's kind of like a telephone wire where proteins in our bodies can use this DNA to signal to each other using electrons. And it just kind of blew my mind. And so that was what really motivated me to work with her as a grad student.
That's a really cool way to think about it. And I think the metaphor still helps. I think that's amazing. So then when you were getting close to the end of your PhD, this is another kind of big transition or pivot point for a lot of people. How did you decide you wanted to pursue a postdoc, first of all, rather than go into perhaps industry? And then how did you decide to go to University of California, Berkeley?
Yeah, those are great questions. And I think a lot of it has to do with advice I try to give everyone, which is you should make decisions that are right for you at a certain time. So my husband and I actually met in grad school. We were in the same lab. He was a couple years ahead of me. And so he had graduated and started a career in industry in the Bay Area. So I knew that, I really wanted to pursue a faculty job, but, I also wanted to not be long distance with him forever. So I specifically looked for postdoc positions in the Bay Area. And I think sometimes that's kind of seen as taboo in science to make decisions that aren't based solely on the best choice for your career. But, I just want to emphasize that that was the right choice for me to eventually get to where I wanted to be because it enabled me to have my support network.
Absolutely. And I think that is a really important point to drive home, to really think about your life situation as a whole when you're making these different career transitions. And then you made another career transition in 2019. You moved all the way across the country to MIT. What was that experience like, and what ultimately drew you to MIT?
Yeah, that's a fantastic question because I think the reputation of MIT is, well, first of all, it's an intimidating place. And I do like to tell students and postdocs when they're visiting and deciding if MIT is going to be right for them that I was terrified when I came here for my interview. I had heard all of these things about how it's a really unforgiving place and, you can't make mistakes or be yourself. And that's just not what I've experienced at all. What I found during my interview and afterwards is that everyone here is incredibly supportive. And so what I try to get students to understand, especially if they're, deciding whether to come here for grad school, is that the only way that you won't get to experience MIT is if you don't apply. So I encourage people to apply and not kind of self-select out. It's an amazing place and it's really not like any other university. What really drew me here eventually for my job is that they do care about the translation. So I think a lot of times we as professors can get very wrapped up in our lab experiments and forget why we need to care about what the impact of our research is. And MIT does a good job of making you kind of step back and realize that the goal is to really make new things that can actually help people. And the students that come here are students that really want to spend their time doing things that they can see the direct impact of. So I've really enjoyed it here so far.
Absolutely. And I think the research that you're doing there in your lab at MIT has tremendous potential to help people. So I'd love to talk about some of your current projects. Is there one in particular, Ariel, that you are working on at the moment that you are just the most excited about?
Oh, that's a tough one. Let me just start by saying that all of the projects in our lab are driven by our students and postdocs, and they are exceptional. And so having a favorite project is like having a favorite kid. I just don't have one.
Mm-hmm.
They're all amazing. And so I'll talk about one of them because I think it's really great and a good example of how we think about science in a way that kind of takes it back to the basics. So this is a diagnostic that we started out looking at for HPV, human papillomavirus, which causes about 98% of cervical cancer. So it's actually a virus that causes cervical cancer. And the problem is right now that even in developed nations, the main method of detecting HPV or cancerous transformations are pap smears. So it's an invasive procedure where they scrape cells off the lining of the cervix and then visually inspect them to look for kind of cancerous transformations. And it's even worse in developing nations where the standard of care is what's called acetic acid treatment. So they basically take concentrated acid and directly apply it to the cervix and then look for lesions that basically turn a different color. So you miss a lot of these precancerous lesions and it's painful for patients. They don't want to do it. And so a lot of these cancers are missed or misdiagnosed. So there's about 4 times the rate of cervical cancer and eventual mortality in developing nations compared to developed nations. And that's just hugely problematic for me because we have the infrastructure to better monitor these infections and eventually cancerous transformations and, intervene sooner. So I have this really amazing postdoc in my lab, Marion Milligan, who was out to dinner one night. And when dessert came, there were these little flecks of gold leaf. And we use gold electrodes in our lab, and they're generally pretty expensive. And she was thinking, well, if this gold is inexpensive enough for them to put on dessert, it's probably inexpensive enough to make electrodes out of. And so she developed this way of making gold leaf electrodes that is basically arts and crafts. So normally you need to go into this fancy clean room and do a lot of fabrication with expensive equipment. We use a scrapbooking cutter plotter to cut out little pieces of gold leaf, and we put them on transparency film. And so we use about 16 cents of gold, and these electrodes cost us about 50 cents. And then we can then use biochemistry to specifically detect these HPV infections. And so we can do this whole workflow for about $2 in our lab. And we're currently working to make it into, a device that kind of looks like a COVID test. So you can do this at home.
Oh, that is remarkable. So where are you on the timeline for this project?
So we are actually at the point right now where I have a grad student who's prototyping little cartridges for eventual commercialization. We are working with a couple of different companies and a startup to hopefully move this out of the lab and into a company within the year.
Well, that is absolutely amazing. And like we alluded to, such a tremendous potential for impact, particularly in these developing countries. And I think being able to move this, not just, from a cool experiment in the lab into the hands of people is a huge step in science that you alluded to often gets missed. So do you think this is something where you're going to see the project all the way through, or do you anticipate be partnering with other sort of biotech and other companies to move it to that stage?
So we like to be hands-on as much as we can in that process. We've moved a couple of technologies into either established companies or startups from my lab. And I think especially my students are very motivated to learn what that looks like for their kind of long-term career progression. So they are very hands-on in a very productive way in terms of, having really interesting and meaningful conversations with these companies very early on. And I have to say the types of questions they ask are just so much more insightful than I would have ever been able to ask when I was at their stage. And it's really incredible to both see their growth as they think about their projects and what needs to happen in order for them to become commercially viable while also getting to interact with these companies.
I think that is so exciting and wonderful to hear that this is progressing along that translational pathway. But I know there are challenges that you encounter along that pathway and just general challenges in science that you face every day. So do you have a favorite story, Ariel, of a challenge or a struggle or a failure that you've experienced that you could tell me and our listeners about and maybe talk us through how you got through that tough time?
Yes, definitely. I actually use this as motivation for many of my grad students. So when I was a graduate student, I had, I think, 4 projects that I was working on my first year. And I was chugging along and nothing was really working. And during my second year committee meeting, my committee basically threw out all of my projects and said I needed to start over because they didn't think any of them were going to work. And so I left that meeting pretty defeated because it was 2 years of work that I saw as just kind of being useless. But what I found was that, even though those specific projects weren't what I continued with, I was a much better scientist for having worked on them. So, it took me a while. It was a very frustrating couple of months, but, everything I did after progressed much faster, and I was able to develop control experiments and things like that much more quickly and efficiently than I think I would have otherwise. And it really taught me to fail fast, which is one of the things we think about in our lab. If you can run an experiment to see if an entire project has any potential of working, run that one first, because you'll save yourself a lot of time and energy and stress. And, along with that, what that meant was I didn't publish my first paper from grad school until my 4th year. And you can see that I have had a successful career from there. So I try to tell people to not internalize the struggles of grad school, especially when their friends might be publishing before them, or, everyone's PhD looks different. I think it's really about how you handle the hard times that, will kind of define what your PhD ends up looking like.
Absolutely. I think you brought up a really good point that those people that sort of luck out on their first project don't necessarily get to experience and learn how to bounce back after some of these failures, which will happen in your career, even if it's not during your PhD.
Yes, definitely. And I think, honestly, this is the biggest piece of advice I try to give students thinking about grad school is just the number one thing you need to be a successful grad student is resilience. I think a lot of students go into grad school expecting it to be more like undergraduate work, where if you work hard enough, you can get a good grade, and that is kind of linearly correlated to your success. And in grad school, it's not linear. A lot of times you don't really have control over the outcome of something. You can work really hard on an experiment and have it not work. And it's not a reflection on you or your abilities or your science. So I think just kind of internalizing that and being able to bounce back when things aren't working is the biggest thing that you need to be successful in grad school, and kind of something that I think is not necessarily emphasized when we explain how grad school works, especially to undergrads.
Absolutely. And I have to ask, Ariel, was there ever a point during that maybe dark period of your second year of graduate school where you thought about leaving science or thought about changing your field or sort of making a big pivot?
Honestly, no. I'm very motivated by tough times. And so, I wanted to kind of prove to myself that I could be successful in spite of these challenges. And so I think one thing that was nice for me is my committee did give me a lot of freedom in the kind of projects I picked up next. So I got to kind of reshape what my PhD would look like. But after having 2 years of experience hearing about all the projects in the lab and getting a little more comfortable with the science. So, in retrospect, I think it was a really incredibly beneficial opportunity to be able to kind of redo my first year, but with the experience of a second and third year.
Well, Ariel, I know you were able to turn this struggle around, and you've had many exciting successes since then. And listeners, if you want to hear more stories about exciting scientific successes and discoveries, New England Biolab's podcast, Lessons from Lab and Life, is your source for staying informed about the most up-to-date molecular biology breakthroughs. Host Lydia Morrison interviews prominent scientists and delves into their research to uncover how their findings affect society in meaningful ways. Tune in to learn from the experts and get a glimpse of the cutting-edge advancements being made by scientists around the globe. Don't miss out on this opportunity to gain new knowledge and stay informed on the latest discoveries. Tune in to Lessons from Lab and Life today, wherever you listen to podcasts. And Ariel, in terms of successes here, do you have a favorite success story of your own, whether it was a big win or even a small but particularly meaningful victory for you?
One of my favorite things to talk about is one of the platforms we've developed in my lab is we can take these microbes and we can put proteins on the surface, and we can do that genetically. So you don't have to chemically attach them or, isolate and purify proteins. And a lot of people are like, how did you come up with this as an idea? And it was really just laziness in my postdoc. I didn't want to have to purify proteins. And so I did some reading and figured out ways to get around having to isolate and purify proteins. And it has turned out to be beneficial in many more ways than just not having to purify proteins in terms of, the stability of things we can put on the surface and the kind of breadth of projects we're applying it to. But, it really was founded on this pretty simple concept of not wanting to spend my Saturdays at a FPLC or a machine to purify the protein.
I love that. I think sometimes the easiest solution is the best solution. If you can avoid some of the struggles and the pain, absolutely do it. And this idea of applying different things to the outside of different bacteria and organisms has really expanded in your lab. And I'd love to touch on just some of the applications and what you're doing with this sort of big picture idea.
Yeah, definitely. So we kind of have two different ways we think about, microbial surfaces. One is we genetically put these proteins on the surface, and we're using those for a lot of different applications, mostly in environmental remediation. So we have microbes now that can degrade pesticides that kind of look like chemical warfare agents. We can degrade forever chemicals, PFAS. We can degrade plastics, and we can even use them for recycling. So these proteins on these microbes are much more sturdy than they are in other situations. So we can, recover rare earth elements or other kind of precious metals from acidic conditions or things that are normally kind of inhospitable for proteins. And so we really see it as a platform where if somebody has a contaminant they want to try to degrade, we want to work with them to try to figure out how to degrade it. And then we have a complementary method where we're trying to actually use chemistry to protect microbes. So this is kind of the inverse, where we chemically coat microbes, kind of like forming a hard eggshell, and then that protects microbes during manufacture and processing. So we can use microbes for a lot more applications than we could before.
Absolutely. And I'm particularly fascinated by this idea of sort of building a shield around these microbes. I think there are many different applications, and one of them that you recently published on is related to using microbes in fertilizer. So can you talk a little bit more about this project?
Yeah, definitely. So something that has really motivated me. I'm originally from the Midwest. So I'm from St. Louis, which is in the middle of kind of corn and soybean territory, and seeing kind of the impacts of agriculture, the fluctuations in pricing and everything, even just on the economy, really made me interested in learning more about what farmers need. And what I learned pretty quickly was that fertilizer prices are very tied to oil prices. And it's very expensive for farmers. And so, this project, again, was kind of motivated by giving people access to technology that they didn't have before. So chemical fertilizers are not only expensive, but they're pretty bad for the environment. Agriculture is about 11% of greenhouse gas emissions in the US. And chemical fertilizers people apply generally about 30 to 40% more than they need. And the rest ends up as runoff that can damage local environments. So not only is it wasteful, it's also damaging, and it produces greenhouse gases. So the alternative that we like to think about is what's called regenerative ag. This is a step beyond kind of organic farming, where you actually put microbes back in the soil that are supposed to be there, but most of them have died off because of chemical fertilizers. And using those microbes, you can basically replace all agrochemicals. So we have microbes that can do the same things that pesticides do the same things that ammonia does, do the same things that phosphate-based fertilizers do. And so if you have the right mix of microbes, you can basically remove the need for chemicals. And that's fantastic. But a lot of these microbes are really delicate. So the reason people aren't all using microbes right now is because we can't manufacture them or transport them. So what we found is that our coatings allow you to manufacture and transport temperature-sensitive microbes at very high temperatures. So this allows, farmers in places like Brazil and India to store these microbes. We found that even these microbes that were stored at 130 degrees Fahrenheit, which is 50 degrees Celsius, they actually improve germination over live microbes. So we can take these dried, stressed microbes that we put our materials on and get about 150% more germinated seeds than with fresh microbes. So we're very excited because the materials that we use to coat the microbes are all generally recognized as safe. And so it's like a fancy FDA term for just being food safe. So we're not adding any harmful chemicals. We are, improving the yield of these processes. So that's also indirectly reducing emissions associated with their production and, negating the need for all of these agrochemicals that have a lot of harmful side effects.
This work is fascinating, Ariel. And you mentioned, all of these benefits, all of the positive things that could come out of these becoming sort of more broader scaled out into the world here in the agricultural community. What are the limitations or kind of what's keeping you from this next step?
So actually nothing right now. We spun this technology out into a startup called Scialog. So we're a pretty small startup right now. We founded about a year ago, but our goal is to work with companies that are engineering microbes to make them more efficient as biofertilizers to help them scale to manufacture by using our materials.
Oh, I think that is remarkable. And you mentioned that this sort of startup side and this idea of spinning companies up out of the lab is something that your group has been doing. Was this something that you knew you were interested in before you started working on these particular projects?
Honestly, no. We went through a lot of kind of the training that MIT offers for entrepreneurship. And that was what really made us interested in this. I like to joke that, we have very, very good undergrads here. So please know that this is in jest, but that the reason we thought that this project would be such a successful startup is because freshmen can come into my lab and never having worked in a lab before can coat microbes successfully on their first try. So if it is that translatable, it just, helps with scaling and everything.
Absolutely. I think that is phenomenal. I think this path is something that particularly in recent years, a lot of undergraduates and sort of trainees in general are interested in sort of launching into the startup sphere. So it's wonderful to hear your perspective and how that experience has gone for you. And we've talked about some different pieces of your career. I think one of the things we haven't yet touched on is travel. And I think this is a big part of the life of a scientist. You get to go to a lot of places for conferences, train in different places for your different stages of your career. So Ariel, when you think about all of the places that your science has taken you, do you have a favorite or maybe most memorable place that you've been to?
I think the most inspiring place, I'll say, I got to go to a science and technology conference in Kyoto, Japan. So Kyoto is amazing. And it was really incredible to get to spend a few extra days there kind of touring around. But the conference itself was a mix of scientists in academia, scientists in industry, and policymakers. And I think one of the best things that I've gotten out of being able to travel is to meet with policymakers and understand how they look at problems in a way that's very different from how I do.
Oh, that's interesting. So has that helped you kind of shape how you tell this story now that you're back in your lab?
Yes, definitely. And I think it also helps us figure out what sort of, either metrics we need to hit or, what would be a success for policymakers, which is, very different from what it would look like for industry and very different from what it would look like for my lab. So kind of getting that complete picture is very new for me and really exciting.
Absolutely. I think these conferences where they bring lots of different stakeholders with different backgrounds together are the most fruitful, right? Because you get to have these conversations that you aren't really able to have necessarily in your lab there at MIT, where you're surrounded by other scientists most of the time.
Exactly. Yeah, definitely. Completely agree.
And you mentioned that you were able to add maybe a couple of days onto the trip to explore Kyoto. Were there particular things that you were able to do or see that you recommend any person traveling to Kyoto add to their itinerary?
I very much enjoyed going to the temples there. They are incredibly beautiful and shockingly different from one another. I think I had seen pictures of some of them and kind of gotten a sense of the architecture and had wrongly assumed that they would all look pretty similar. And that is not true at all. And each one kind of has its own goal. So what the temple is meant to kind of be for, whether it's, monetary success or good health or relationships. And they're all so beautiful in such different ways that it was, a really fantastic experience.
Well, that sounds lovely. Listeners will definitely have to tour all of the temples of Kyoto if we're able to make it over there. And we talked about the people that you've been able to work with in science. And I think these can also be phenomenal assets to the career. I think being able to work with people who are these fun, creative people is not necessarily what people think of when they think about careers in science. I think the media has portrayed scientists as these very serious, austere people working in a sterile lab environment wearing their white coats. And I think that's just not the case in so many of the people that I've met. So do you have examples, Ariel, of maybe quirky traditions or just fun or funny stories from you and your colleagues that you could share that really show this human side of science that cuts against some of those stereotypes people may still have?
Yeah, the first one that actually came to mind is in grad school, we had a softball league within Caltech. And, some of the labs definitely took it more seriously than we did, but we had a 5-year losing streak. So my entire time at Caltech, we didn't win a single game, but we went out and played every week. And it was so much fun because, people would bring their friends and significant others. And, there was always beer at 5 p.m. And it was just a really nice kind of way to get to know your lab mates outside of the lab environment. And I think that we've tried to kind of continue those types of traditions in my group. One of the first activities we did together as a group when I started was going axe throwing, which is amazing because they made everyone come up with an axe throwing name.
Oh my goodness.
I know. And we have one of the boards ended up falling down during it. And so they let everybody sign it and I have it in my office. And it's my favorite thing to explain to people when they come to my office.
So Ariel, what was your axe throwing name?
Oh, I don't even remember what mine was, but the person who won was the very soft-spoken postdoc who took the name Bone Crusher.
That's amazing.
So Bone Crusher is written in very large letters across the plank.
Well, that is an amazing tradition. And I love to hear that you and your lab are able to kind of get together to have fun outside of the lab, because I think you're absolutely right. That brings the group together, helps people get to know each other, and sort of helps build that sense of community that I think is critical to solving big problems in science, which you're definitely doing in the lab. And I know oftentimes what keeps you from addressing these big questions are small details, right? Like funding, staff, technology, feasibility, time, all of these minor inconveniences in science. So Ariel, if we took away these barriers and gave you unlimited resources, what is the one research question or problem that you would want to tackle first?
Honestly, I think that we're addressing all of the research questions that I think are the most important right now. I think what I would do if I had unlimited funding is make sure that all grad students and postdocs had equitable pay kind of across the country.
Definitely. I think this is a huge problem and something that's come up a lot in the media, particularly lately, where people are trying to change this. So what do you see as the biggest barriers to successfully implementing some sort of equitable pay system?
Honestly, I think it's just lack of general understanding of how grad student and postdoc pay works, because I think, generally grad students and postdocs, unfortunately, think that, we as faculty have a lot more control over it than we do. If I was going to kind of call to action listeners, it would be ask the government for more funding, because until we get bigger grants, our hands are kind of tied in terms of salaries and things like that. And I definitely know that both grad students and postdocs are underpaid in academia. And so it has to start from kind of the fundamental funding agencies and then move through the system.
Absolutely. And I think in many cases, not only sort of the limitations in salary is something that people experience when trying to go through and sort of survive graduate school and postdoc days, but also maybe or maybe not being able to join a lab that they want to because there's just not enough grad student slots or postdoc slots on a particular grant. So I think that's another limitation where it limits the opportunities of people who are interested in training on these particular projects, but also the ability to sort of execute these bigger scale projects as well.
Yes, I completely agree.
Well, I think this is a worthy cause for unlimited resource application. So we appreciate you dreaming big with us on the show today. And Ariel, I'd love to wrap up our conversation today by talking about advice. So do you have a piece of advice that somebody gave you at some point along your career path that really helped you that you can share with all of our listeners today?
The best advice I've gotten and now given is that every single piece of advice and every single thing I've talked about for, the entirety of our conversation has been within the framework of my kind of survivor bias, right? I'm a faculty member at a place that I really enjoy working. I have an amazing lab that's doing really fantastic science. And so if anything that I have said throughout this doesn't resonate with you, just disregard it, because I don't think the same path works for any two people.
Absolutely. I think that's wonderful advice. And I think it sort of comes across when you even go to your lab website, you get this feeling of you really support diversity and inclusion and thinking about everybody's different perspectives. So maybe could you comment on how that's such an important element of your group there at MIT?
Yes, definitely. I think both within my group and within the chemical engineering department, I have really tried to improve how inclusive things are, because I think the first step to more diverse groups of students, even just in terms of their scientific backgrounds and how they think about science and approach science, is just making sure that everybody feels like they not only have a seat at the table, but that they're invited to the table. So my goal is to make an environment where everyone feels that, and to hopefully have that translate to much better science. I mean, I think at this point, the research has shown that the more diverse a group is, the better science they do. And I think we are kind of living that. And I'm hoping to kind of continue that, as we have a lot of undergrads working with us from very different backgrounds and very different, even departments within MIT. We get material science students, grad students, and undergrads. We get mechanical engineers. We get everybody. So I think that's really my goal.
Well, Ariel, I am so glad to hear that you are cultivating this kind of environment in your lab. And I think you've shared so much wonderful information with all of our listeners today. So I'd love to wrap up with one last maybe piece of motivation or inspirational note, if you have one.
I think my main kind of advice or motivation is make sure that you make the time and the space for yourself. So the main thing that's going to make you a successful grad student or postdoc or scientist or, researcher is your mental and physical health. So that should always come first. So make sure you're in an environment where that is honored and supported.
Well, such an important message. I think wonderful note for us to end on here, Ariel. If our listeners are interested in what we talked about today and want to learn more about you and the work that you do, what is the best way for them to do so?
So we have a lab website. It's just firstlab.mit.edu. And we also try to keep everybody up to date on what's going on in the lab on Twitter/X. We have @firstlab, as well as my handle, which is @AFirst1. So both of those will be great resources to learn more about what's going on.
Phenomenal. Well, listeners, definitely get connected with Ariel and her lab. Check out her website to learn more about the amazing work they're doing. And Ariel, it's been such a pleasure to have you on the show today. Thank you so much for your time.
Thank you. And thank you for such amazing questions and discussion. It was really fun.
Well, it was a pleasure to chat with you and learn more about all that you're doing. And listeners, always wonderful to have you here with us as well. We'll see you next time on another episode of People Behind the Science.